We're in Lab 5, Passive Transport Lab. In this lab, we have 3 different activities. The first one is diffusion. So we prepare 2 different kind of dye to watch them to diffuse in argon. Argon is like water. It's 99% water. It's like the gel you purchase at HEB. So it's mainly water. But here's some fiber so it can slow down the diffusion. And in the class, we talk about diffusion. Several factors will affect diffusion. And molecular weight is one of them. So bigger molecule gonna run slower, basically. And this time we prepared 2 kind of dye. So one smaller, one bigger. And we put them on the argon. And you just watch them diffuse out. So which one gonna run faster? And of course, the potassium permanganate, that's the smaller molecular. Molecular weight is only 158 gram per mole, compared with methylene blue, that's the 320 gram per mole. So you watch them, and after 10 minutes, 20 minutes, 30 minutes, well, you're gonna find the smaller molecular weight diffuse out more. So diffusion rate, molecular weight will affect the diffusion rate, and you can watch this video. It will show you how they change through times. And there are also other factors. Right? All these factors will affect the diffusion rate. The second activity, osmosis. So osmosis is water movement. And we use egg to do the experiment. So the egg, we put them into different kind of water. So first they need to do the process, remove the shell. And then they have the egg and covered by the membrane. Egg is actually a cell. This is the biggest cells we can find. And we put them into different tonicity solutions. We put them into the hypotonic and hypertonic. And in the class, we talk about the hypotonic solution going to make the cell swell because water flow in and try to dilute the solid. And hypertonic solution going to make the cell shrink because water going to move out, try to dilute the solid. So the experiment, after a few hours, we found, well, the cell, the egg put into the hypotonic solution actually become bigger compared with the one in the hypertonic solution. So you can watch this video. It will show you what's the response. And experiment 3, we use the artificial blood. Well, we won't do the experiment at home, but I can explain the experiment to you. So it's pretty straightforward. We prepare 2 artificial blood solution. So these are the process. And in this experiment, you learned, okay, what really decides the tonicity? We talk about hypertonic, isotonic, hypotonic solutions. And the reason the water have to move is the water move to dilute the solute. So if the solute can move, then the water does not have to move. And we calculate osmolarity. This one teaches you how to calculate osmolarity. So you take the molarity and multiply the number of particles. That's the dissociation factor. That becomes the osmolarity because we consider how many real particles you have in the solution. And also, the key point for experiment 3 is only the non-penetrating particle will decide the tonicity, because only the non-penetrating particle the water need to move to dilute the non-penetrating particle. So the tonicity only been affected by the non-penetrating particle. If the particle is penetrating, then water does not have to move. So we talk about isotonic solution, hypotonic solution. It's all based on the non-penetrating particle. How many non-penetrating particles you have in the solution. So if you have way too many, the water have to move out to dilute, then it shrink. If you have too little, the water move in and the cell swell or hemolyze. So it's all based on the non-penetrating particle. So here are 2 examples. Okay, if the outside you have 500 milliOSM sucrose, inside is 300, and compare with 500 milliOSM urea, and inside is 300, even though the concentration are all the same, this is non-penetrating particle, this is penetrating particle, and the results are totally different. That's the purpose of this experiment. So let's look at this one. So outside has 500 milliosm sucrose. So you put the red blood cell into this solution. And while outside is high concentration, and this is non-penetrating particle. So this particle, it want to move in, but it could not. So the only way, only thing can change is water. Water gonna move out from here to here. Water gonna move out to dilute the solute. So it turns out the cell gonna shrink, and that's crenate. Crenate is a big word to say the red blood cell shrink. But if you put the red blood cell into a penetrating solution, like 500 milliosm urea is penetrating particle, and what's going to happen? The water does not have to move out because urea is penetrating particle. Urea just flow in. Every molecule want to go from high to low. Urea is high outside, low inside. It's going to flow in. Especially it can flow in. It will flow in. So the water does not have to flow out. It's the urea flow in, and water actually follow the urea flow in. So the cell swell and hemolyze, and that's the purpose of this experiment. So we prepared two artificial blood, and they're the artificial red blood cells, and we have two test tubes, and we add sucrose, add urea, and we wait. Okay. 10 seconds, 20 seconds, 1 minute, and quickly you found, okay, well, the one with sucrose one still look like blood. But the one with urea, it quickly become clear. Clear is hemolyzed cell. Why is clear? Because all the red blood cells were gone. And they were gone because they burst. So the third experiment tell you for the tonicity, only the non-penetrating particle will affect tonicity. So I put some practice question for you. I asked you, okay, what solute can enter the red blood cell? Penetrating, non-penetrating. We know the penetrating one can enter. And if you put the red blood cells into isotonic, the cell volume remain the same. And you put the red blood cell into a hypotonic. Hypotonic means the concentration is too low, so the water gonna flow into the cell, make the cell swell. So the cell volume gonna increase. And you put the red blood cell into a hypertonic solution, the water gonna flow out to dilute the solute. So in a hypertonic solution, the cell's volume decrease, crenate. So the tonicity conclusion, tonicity is affected by non-penetrating solute only. Okay, so that's the lab for today.